Assalamu alaikum. Hi everyone. Sir, am I audible?
Yes, you're audible.
Okay, thank you so much, sir. And first of all, welcome all of you, uh, all national and international participants in today's webinar.
First of
And
all, welcome all of you.
entitled Exploring Bioinformatics, Beginner Window to Data and Biology. Here, Dr. Saad Sarfraz, uh, principal investigator and assistant professor at University of Agriculture, Faisalabad, Pakistan, Center for Agriculture, Biochemistry, and Biotechnology. And I would like to invite Dr. Saad Sarfraz for today's webinar. Sir, please go ahead.
Okay, I hope everyone can listen to me. Please, Mansoor or Murtaza, kindly confirm that am I audible to everyone?
Yes, sir. Your voice is audible.
Thank you. Okay, so first of all, thanks for inviting me to this very dynamic group of student researchers, academia, so that we can connect at different stages to explore the window of bioinformatics. Today, we are going to talk about how the biologists or different people working on different fields of life sciences are can benefit from different aspects of bioinformatics. So before starting or jumping ahead, I will just start about brief introduction about me, just to get a bit connection with you guys. Basically, I myself is a from very start I really like genetics, and for that I found that there is a university called as Arid Agriculture University, and they are offering some degree program called as plant breeding and genetics or something like this. So I started my BS from that, and then I joined a local institute called as NIPG. From that I basically did my masters in biotechnology and molecular biology, and then on using Marie Curie fellowship I joined a prestigious institute, Seva, in France, in Montpellier, and this institute offers
Sir, your mic, I think, off. Please check.
Sir, your mic. Okay, so sorry for the disappearance. There was some problem with my internet connection, I think so. So I will start again. Sorry. I apologize to everyone. Okay, so basically, I was here when I left due to some internet problem. So basically, today is we are going to talk about bioinformatics, and the basic bioinformatics is that how we can get simple data into biological information. So these days there are major disciplines and domains. Uh, Normally we call them as omics. Omics has four or five major disciplines. Number one is when the information is present in the form of DNA, it is called as genomics. When we are getting the information in the form of RNA, it's called as transcriptomics. And if we are getting information from amino acids or protein sequences, this is called as proteomics. And after that, these proteins provide some primary and secondary metabolites, and study of those metabolites is called as metabolomics. So these are the four major uh, fields of bioinformatics. Not only that, we have fields like another domain which is called as drug discovery. Bioinformatics is helping a lot in the development of novel drugs that were not known before or present before. And structural bioinformatics, how protein-protein interaction, protein-ligand interaction is unveiling us important information in curing diseases like cancer. And secondly is system biology, how computation, technology, and biology are basically interconnected to find different systems biology, how different systems are actually working together. And the last but not the least is the advent of artificial intelligence and machine learning to perform intelligent bioinformatic operations. So pretty much these are the major domains of bioinformatics. Maybe I missed few of them, but I precisely selected them so that I can help you and guide you about the major discoveries that are happening in the field of bioinformatics. So basically, it's all started when we first able to sequence the whole genome of human being in 1953. Actually, it was started in 1866 when Gregor Mendel made those crosses, and in 2003 we were able to sequence first ever human genome. So that was the biggest milestone. So now we have the information about the coding sequences or non-coding sequences of the human genome. From these human genome, actually things starting to get heat up. We try to find different connections or different story. Today, I'm going to tell you a very interesting story. Maybe you already have heard about it. That in the cave of Denisova, this cave basically is present in southern Siberia in Russia, and they found an interesting sample of a pinky tooth a bone from these caves. And the Max Planck Institute of Anthropology, evolutionary anthropology, basically collected those samples. And we wanted to find out that what kind of human being was that. And we were pretty much sure that it will be a Homo sapien, a modern human being. As you are aware that modern human beings have two species. One is called as Homo sapien, another is called as Neanderthal. So we were pretty much like that there is some people were living inside this cave. They are belonging to Homo sapien. Scientists, what they did basically, they took the DNA from the tooth and then they extracted the DNA and then they will develop the whole genome sequence from that individual. And they find out that basically that it is not the modern human beings, but they have discovered a new species of human beings that is closely related to uh, the Neanderthal, which is and they named as Denisovans. So these Denisovans now have been discovered as the new species of human being just because of the use of genome sequencing and data analysis. So far, we knew that there is another kind of human species was living inside this cave, but we are not able to find out what it actually looks like. So we wanted to find out what is the facial structure, what is the shape, what is the color, what are kind of different kinds of facial structures or bone structures are basically depicted in this human being. So these kind of informations are probably provided by only thing which is called this transcriptomics. So since these species are not no more present today and only we have Homo sapiens today, so it's very critical that how we can get an image to this information. So with the time, we find out more samples. For example, the Lanzhou University discovered new samples. The CNRS Paris, France, basically discovered new samples. So using those samples, scientists basically what they did, they developed an epigenome. So what is epigenome? That this, for example, this is our DNA, and we know that DNA is only not stranded like that, but it has some methyl groups. Uh, and these methyl groups basically provide us the information whether certain genes can be expressed or certain genes cannot be expressed. So basically, they are the regulators that how these genes are expressed with it has whether they have a minimum expression or whether they have maximum expression. To these kinds of information is can be collected from epigenome. So from that sample, we were able to reconstruct the methylation map of these Denisovans and of course Neanderthals as well. And we found that basically Neanderthals and Denisovans both are related to each other. So both have elongated face, they have robust jaws, they have wide pelvis, large rib cage, and wide fingertips. So we were able to reconstruct using those genomic and epigenomic information. We were able to reconstruct the image of that person. So we found that basically she was that person living in the cave was actually a 13-year-old girl, and she was living inside that cave. And when extinction happened, we collected those samples. So in this way, the genome and epigenomes are basically getting us information and getting us past and giving this information from some few thousand years ago. So far today, we were able to sequence a lot of genomes so far from different kingdoms: animal, plants, fungi, protists, bacteria, and archaea. And you can see here, for example, in case of animals, we have covered so far 592 species. In plants, we have got big numbers, 1,667 species. And of course, in bacteria, we have got so many species sequenced now. So we have a lot of biological knowledge now has been converted into data. And we can basically, as a bioinformatician, can get as much as information as possible. So before that, we were interested in reference genomes. This is called as reference genome, sequencing one species. But now things have been changed. Now we are living in the era of pangenomes. So what are pangenomes? I basically like this event to tell you about, educate you about a bit of pangenome. So but basically what is pangenome? Traditionally, we were able to sequence one genome at one time. Okay, but one sample from one species basically does not represent the variation in the whole species. So what they did basically that if you can collect, for example, here we have three organisms, organism one, two, and three. So they must have some core genes which are common to them, and they will have some unique genes that are unique to them. So what if rather than providing a single reference genome, we can provide a genome which has the core genes as well as the unique set of genes. So in this way, we can cover the variation of the whole species in one file. So that is the basically objective of pangenomes. So you can basically analyze many genomes from the same species. You can capture full intra-species diversity, and you can have suitable for trade discovery, evolution, and disease resistance, and you can reduce the preference bias. For example, here is the pan-genome demonstration for human genome. For example, this is our human genome, above represented as a blue line, and at the bottom you can see that they have collected 19 individuals from European ancestry and find out that whether they are linked with each other or not, whether there is a variation or not. So at the bottom you can say, so you can see that you have different variations that are present in the genome. Someone has inversion, someone has single nucleotide variations in them. So these kind of or also duplication as well. So this kind of graph image is actually called as pangenome. So I'll give you a brief example about soybean. So recently they were able to reconstruct or resequence 2,989 NGS sequence samples. So they have now how many seeds they have collected? So around about 3,000 accessions of soybean. So they wanted to develop a pan genome. So what they did basically, they first of all made a phylogenetic tree of these using that resequencing data. And using that resequencing data, they find out that there are 26 representative accessions. So means like that there are the 26 most important ones that actually representing the whole diversity of soybean genome. So what they did basically, they performed the whole genome sequencing, total genome sequencing, using different using PacBio specifically and sequence it, and they find out that in total, all the soybean accessions that are present in the world has 20,623 core number of genes, and 57,492 uh, those genes that are not common to all of them. So then they developed the synteny, then they studied the structural variation. So this is actually the representation of graph-based genome that is shown here. So these are the applications of a pan genome. For example, first of all, on the left side, you can see that it basically tells us that whether a gene is present or absent, present absence variation. For example, at the top, at haplotype 2, the gene HPS encoding gene is present in haplotype 1, but it is absent in haplotype 2. So you can see there is a difference. Then you can also study fused gene. For example, in these first two genotypes at the shown at the top, they have actually uh, they don't have a third gene called as they indicated as other gene, but now in the third organism or the third species or third accession, this gene is got fused. And also we can study the evolution, what kind of structural variation is present, if there is kind of some kind of structural variation, the inversion, duplication is present, and also we can see the expression differentiation. We can take the RNA from different species and see which genes are expressing in a different manner from one species to another. So in this way, one file is basically giving us the information about 2,898 accessions of soybean. So currently we are in the world of pangenomes, and these pangenomes are helping us a lot in introducing desirable traits in our species. So, what are the other examples? For example, we can improve the crops, we can improve the human health, we can discover population specific variants affecting disease or drug responses, we can study antibiotic resistance, pathogenicity, and of course, we can study the genome dynamics and speciation. Okay, so now basically we are at this stage that the researchers from Yale University is actually they are developing a pan genome for human beings. So basically they are collecting DNA from different ethnicities, and they are going to develop a pan genome or super pan genome, you can say, for human beings. One file that has the whole diversity of Homo sapien. Isn't it interesting? So interestingly, we are living in a very interesting time of human civilization, that we have so much information at our grasp. We can do wonders with this information. So that is a bit of flavor about genomics. So once we have covered the topic of genomics, and now the human beings actually want to get attention towards proteins. So the most important thing that you are getting from the genome is actually proteins, but how these protein structures are actually depicted is still is pretty much unresolved. Before prediction, we were used to do predictions or something. It is very difficult to get the actual structure of protein. So whenever scientists want to deduce a protein structure, it takes time and resources. There are basic steps. For example, you have to express protein, and then you have to crystallize, then you have to collect the data about it, and then you have to find out structure determination. If you go step by step, you have to express your protein and isolate it, then you will crystallize for X-ray crystallography. And to perform X-ray crystallography, either you can perform NMR spectroscopy, or you can perform cryo-electron microscopy to study how basically the protein has arranged itself in different structure, so that you can see what is the structure determination. And then you have to, of course, do model refinement and validation. So if you can ask me, it takes a minimum of 6 months to deduce one protein structure. So it is a lot of time. So we are talking about, for example, in case of soybean, we have 20,000 common genes and 50,000 those genes that are variable. So just imagine that how much time we are going to spend if we want to actually deduce the structure step by step. So what they did basically, the scientists developed a tool called as AlphaFold. They use machine learning approaches. So if you can provide a protein sequence, so it will basically dig out the structure library and the sequence library and give you, using training and prediction model, give you actually the structure of your protein. So in this way, without going to basically utilize all these steps, actually you when you put your sequence, you ultimately get in one click, you get AlphaFold 2 structure, which is closest to your already depicted structure. That was the biggest leap after the discovery of the human genome so far. So for that purpose, this team of scientists from Google, Damis, John, and David Becker, basically received the Nobel Prize in chemistry in 2024, last year, basically, and because of their AI breakthrough, and they predicted the atomic level accuracy for 200 million proteins. So if you are interested to study protein structure, what you have to do, you have to go to Uniprot KB over there, and you have to type in your sequence, the name of the gene that you are interested in, and then insert click, and then you will find this window that is represented here, and on the left side you will find a small tab called as structure. And when you click that structure button, you will see that this protein has been already, the structure has already been generated using AlphaFold. And using that information, you can basically perform different kinds of activities. For example, you can perform molecular docking, or you can perform cancer studies, you can find drugs, and you can find a bunch of studies that are possible using that structure. So see, we have so much structure that already been reduced, and for those you have not been reduced, for example, for some plant species, so you can use the AlphaFold and you can generate those structures. So you have one of the biggest source of information at your palm in your laptop. The last part of today's talk is basically, we are in the world of machine learning and generative AI. So there are key applications so far that I'm going to discuss about relatively. There is radiogenomics, cancer subtype prediction, protein ligand affinity prediction, and cell type classification. One by one, I will just give you a brief overview how machine learning and generative AI are helping us in different fields of biological sciences with proven and published studies. First of all, is radiogenomics. What is radiogenomics? So basically, we are getting images using MRI, magnetic resonance imaging, or MRS, and magnetic resonance spectroscopy. So we are getting the image of a patient, for example, suffering from cancer or suffering from any kind of neurodegenerative order, and you get the images or scan about the information. And of course, you have the clinical information from that as well. To combine those graphs, and you can combine the gene methylation and gene mutation differential gene expression together and perform radiogenomic association analysis using artificial intelligence. And then you find out which are the markers or biomarkers are actually covered for that particular cancer. So this is one information, that you are using different scans of your brain, different type of scans, and then you're associating this information with gene expression, and at the end you are going to find out some interesting biomarkers that are basically responsible for that disease. Basically, it helps us to decide or design precision-based medicines. Another thing that it's common that by the help of artificial intelligence, we are predicting cancer prognosis. Cancer is one of the most, we can say, worst disorder the human being has so far have been faced. And the most important problem with cancer is that we are not able to diagnose the stages of cancer, and we normally patients came to know at the latest stage, which is the last four stage of cancer, stage 4, where the tumor is already malignant and it's not being controlled so far. So This kind of model, basically what it does, it takes the information from hospitals and from journals. They have take the information from different hospitals, the scans from different research studies using journals and hospital, and use that information and train a model called as tiger model. So this basically use the information of images and tells that whether this tumor is benign or whether this tumor is malignant. So once the tiger model has been trained, so you can give any kind of data, any physician can give a new scan, it will tell whether the cancer is benign or whether cancer is malignant. Benign means the it is located at one place, or malignant means it is moving from one place to another, which becomes quite complicated to handle. Another thing is that we have discovered, so in the artificial intelligence, is that you might have heard about docking and simulation. So these kind of tools actually tells you that you don't need to perform docking and simulation anymore. So as you have the information of protein, you have the information of ligand, and you give to this model, and basically it basically calculates what kind of interaction it is, what kind of hydrogen bond it will make, what kind of metallic bond it will, hydrophobic interaction or van der Waal interaction it will make, and form interaction network. And from that interaction network, we were able to find out whether you have a positive interaction between a protein or a ligand or protein-protein, or whether you have a non-stable interaction. So this kind of achievements that has been embedded in the case of artificial intelligence allows us to discover much more. So in this way, bioinformatics is helping us in different disciplines. The purpose of sharing all this information was to motivate or entice you about how you can learn from how you become from biologist to a bioinformatician. It's very easy. I did it, and I think all of you can do it. So what you have to do is to, first of all, you have to learn the basic programming. You can use Linux or R. You have to take these two courses which I have lined here: basic command line tools in genomics. It is present in Coursera. And there is another course called as bioconductor, basically that provides training in R. So once you've done that, basically, so now you will be able to handle basic tools like BLAST, cluster omega, maths. You can perform your sequence alignments at using the command line interface, using bash scripting or Linux and Unix. And after that, you were able to complete the second course called as bioconductor. Basically, it gives you uh, the freedom to perform your own gene expression of already available data. You can perform gene expression using RNA-seq. You can find your variants. You can find your epigenome profile using different packages. For example, Seaborn, ggplot2, and dsec2. And the last and the most important one is that once you have done the basic things, you can learn the machine learning. So basic, you can learn the theoretical languages, theoretical background about the machine learning, which is the basics of classification, what is regression, what is clustering. And you can use these kind of already developed libraries, scikit-learn, XGBoost, and TensorFlow, that basically allows you to predict cancer subtype, gene expression, or drug response modeling. And then you have to decide in which field basically you want to go. So each field has a unique set of theoretical and technical skills that you have to master. So for genomics, you have different kinds of skills. For drug discovery, you have to have a grip on the structural bioinformatics. For microbiome and mitogenome, you have to have different kinds of structural, sorry, you have to have different kinds of different kind of sequence analysis. And of course the AI in medicine, that you will use docking plus simulations and different kinds of multi-omics approaches to design your studies. So after that, once you have done with that, so you can go join a particular research group and perform an internship, or go into GitHub repositories and do real projects or some kind of internships to take your skills. Basically, in my timeline, it is like you can do this in 3 to 4 months if you are consistent. And once you are consistent, there is so much biological information that you can harness on. You can publish good work, and you can connect with different networks and try to study different kinds of kingdoms. So that is all from my side. So today was just a basic one, basic window to basically entice you and inspire you about this field of bioinformatics, and I hope we will have a very interesting question and answer session. Thank you so much, all of you, for listening. And if you have any question answer, please drop in the comment, or you can ask directly. Thank you. Yes, over to Murtaza and Mansoor.
And thank you so much, sir, for today's insightful session
Thank you.
again. And now, if anyone have any question, you can share your question in comments. One by one, we will answer your questions. Thank you so much for everyone to join us. And PPT will be shared in group, and attendance also we are sharing in uh, comment box. Comment box. Okay. Yes. Anyone have any question? You can ask. Anyone have any question? You can ask.
Okay, so
Sir, a
we
question
have
is.
yeah, we
Sir,
have started to get some question. Yes, please. Murtaza.
uh, yeah. Sir, how can pangenome improve genome-wide association studies? How can
Yeah, that
pangenome improve?
Yeah, that's a very interesting question. So, Murtaza, the point is like, so I think, funny I've asked this question. That's a very interesting question. So what is genome association study? It's basically we are actually associating phenotype with the genotypic information we are getting. So, what we are right now doing in different labs for plant improvement is taking a set of genotypes, performing genotype by sequencing or short read sequencing data to find out what kind of mutations are there. So as far as we have more mutations, like for example, we have the information about pangenomes, so we know what kind of reference mutations that could be present. So once we have the basic information, what mutations could be present, so these kind of information will ultimately help us in developing different kinds of chips that allows us to streamline this genotyping by sequencing method or short read sequencing method. So the prior information is being provided by the pangenome. So that is the answer to that. Next question, please, Murza. Will you
Thank you
take
so
or
much.
I will go ahead directly?
Yeah, sure. Yeah, sure. Sir, I am speaking.
Okay, please. You can take the next question and tell me.
Sir, next question. R is also great. How is it is about Python for data scientists?
Yeah, that's a very important question because today was a beginner lesson, so that's why I didn't stress too much about Python. So first you have to learn that any kind of biological sciences data, you can of course use Python. If you have skill in Python, you can do all those analysis. But uh, once you are if you are at a starting point, I would suggest that you go towards Linux or R, and then you come at Python, because most of and majority of our tools are basically still present in Linux, and so you have to have the basic understanding of Linux as well. Python is actually best, and Python is basically language designed to perform machine learning or generative AI. So yeah, I think Python can be utilized at the later stages. And for example, if you are interested to develop softwares, you want to write new codes or something, then that becomes quite interesting. But now, I think Unix based, Linux based, and R based script writing would be essential. Next question, Murtaza.
Uh, Thank you so much, sir. Next question is, what is the like scope of bioinformatics? One question.
Okay, so basically, I basically described here already that you can do a lot of things. For example, here in radiogenomics, you can have cancer subtype prediction, you can have protein-like interfinity prediction, you can improve crops, you can do a lot of things. Basically, uh, bioinformatics is serving as a toolkit to address key questions, whether it's plant health, whether it's plant production, whether it's human health, whether it's microbes, or whether it's evolutionary studies. So all of this can be answered using bioinformatics. So what's up? Next question, please.
Thank you so much, sir. Next question. I think no more specific question. If anyone
Okay.
have one question is, so sir, the role of
Yes, yes, please.
AI in the role of AI in changing gene sequence in future. Does AI changes in gene is helpful or not? Like he want to interpret.
Yeah, that's a interesting question. That what kind of information that should be present. Actually, I'm not expert in this field, but there is one field called as synthetic biology, and a lot of people are working on that field. Basically, they are developing new strains, or they are editing the genomes of new microbes to perform essential functions. For example, they want to degrade plastic, they want to degrade important component or pathogen, or they want to degrade some important substrate. So basically, they are editing and making new genes by themselves. So here, the artificial intelligence can help them and help us with one kind of editing can be done to synthesize entirely a new gene, and in this way, you can improve the microbes to solve different problems regarding environmental or regarding human health or plant health. So I hope I answered your question. So if there is any other question, please let me know.
Okay, thank you so much, sir. I think kind of what kind of companies hire bioinformatics and how they have client.
Mhm.
I think it's
Okay,
not most relevant.
right.
Yes,
So
please, sir, go
I'll
ahead.
just answer. Yeah. Yeah. Just remember this. That a lot of different labs. For example, you can find jobs in academia, different universities. All the universities, all the labs require one bioinformatician. Okay, this is now becoming a new normal that they need one bioinformatician. So if you are good at your work and you have published good studies, you can definitely join those labs. Of course, it is quite competitive, but you can definitely go ahead with that. And yeah, apart from that, there are different companies that are hiring new individuals for improving this kind of analysis. For example, recently we met Oxford Nanopore Technology, PacBio. I'm also in talks with Illumina. So there are new people that are being recruited from our disciplines, and in this way, they are getting good jobs in this field. So there is a lot of money involved in this one as well, and it's a race of technology. So whoever gets first will get good opportunities for them.
Thank you
Yes,
so much, sir.
Mithun, welcome.
Next question is why we do the centenary analysis?
Yes.
Centenary.
Okay, yeah. So the basically synteny analysis is when you want to find out the arrangement of gene in one species and the arrangement of gene in the other species is correlated with each other or not, is the same or not. So basically, you want to find out whether there is. So this is synteny here in the panel number one, in which we have gene presence and absence variation, and they have developed a synteny between two haplotypes, so two genotypes. So genotype number two and genotype number one, they have developed synteny, and they found that this HPS encoding gene is not present in haplotype two, but it is present in haplotype one. So basically, this is why we do Synteny, that we want to find out whether there is any kind of new gene, whether there is any gene fusion, or this kind of information that can be get from Synteny. So, most importantly, we are actually addressing different problems. Where Synteny is a very important tool for providing evolutionary studies. Yes, Murtaza, please. There is another question, I think so.
No, sir. I think no question right now.
I think there
So
is one question. Just Murtaza, just let me take one question, which is normally the most important ones. So there is
yeah, yes.
one student. What about those who are planning to shift in bioinformatics? What will you suggest at very first step for those who are familiar with basic tools till docking? Are you going to provide more session? So yeah, definitely with the host of this campaign, we can arrange more session, and you can connect with this kind of. But for example, the first thing I want to tell you that you have to get a bit grip about command line. Okay, once you have got a small grip of command line, then we can do a lot of things. Without that, I think it will be really difficult. There is another question, Murtaza, I would like to take. Sir, I have a question. Which AI modules we are using, like Cloud AI, or are we using specific AI modules? So basically, for solving the biological problems, you are not using those natural language processing tools, for example, like Grok, for example, Cloud AI, or something else. So basically, you develop your own models, and you have to train those models, and using those models, you can basically get good answer. So these natural language processing models that are already available, so they are already in big amount of their big size, and it is very difficult to fine tune or quantize those models. So you have to start with the smaller models, and then you have to train those models and get your answers. Yeah, that's it from my side, Murzo.
Okay, everyone, thank you so much for your time, and kindly make sure to fill the form, Google form, review form of today's event. It's mandatory for certifications, and I will suggest you kindly be connected to this group. Maybe we will in future we will uh, arrange more sessions about bioinformatics and biotechnology related. And soon, sir, I want to announce that we are going to organize a international symposium on AI, artificial intelligence in medical research. So we are going to planning for it. So
Okay,
I
that's
would
very
like to
interesting.
invite all of you. Yeah. So still, if anyone have any question, sir, one question is, if you can see, please,
Yes, please.
how we can earn by using next
Okay.
generation sequencing.
Okay,
These
so
tools are
basically, yeah, that's an entrepreneurship question. That is very important. Once you have the basic knowledge about and basic skills to analyze the genome, so you can of course contact with different people who are working with this one. And for example, you can join different websites like Fiverr, or other platforms, and provide your niche over there. But it's a very long process. Okay, but once you are familiar with this, and you will get good clients over there, and you can analyze your data. A lot of my students are working over there, and they are getting good attention about it. And ultimately, in Pakistan, we have researchers, but I think there is still a very big gap, and we have to fill that gap as much as possible. And you can then provide those services for international and, of course, the national audience as well.
okay. So next one question is the optimists. Please, if
Yes. Okay. So how can we? So the question is, how can we accurately detect, characterize, annotate complex singles? So he's basically referring to multi-nucleotide polymorphism or nested indels in the highly repetitive genomic using short-read sequencing data. Yeah, that's a very very difficult one to ask. Okay, so the point is like when you have want to go inside a highly repetitive genome using short-read, it's a very difficult domain to travel, traverse. For that, you need long read. First of all, the best way is to you must have long read data, and in this way you can get a big picture of your repetitive genome sequences. And once you have that long read, you can start to make use different types of variant calling filters, either using BCF tool or different other kinds of packages. And then using those tools, you can find out which kind of variation, whether it's a multi-nucleotide variation, whether it's a single nucleotide variation. So you will be able to guess that. But using short read, it will be a really complicated one. So I will not suggest. For accurate information, I would suggest you go for the long read ones. Yeah, another question, Musa, I will take that, from where I learned data analysis. So basically, you can learn the data analysis using basic courses that I have indicated here. You can go to Coursera, register over there, and you can basically go to a course called as Command Line Tools in Genomic Data Sciences, and also, there is a course called as Bioconductor over there, and you can use those courses to train yourself in command line. Uh, so these are some courses where you can start to learn the data analysis, and then after that, there is a you can say a learning curve for everyone. It can go smoother or it can go a bit harder, but it will be ultimately better for you. I think we have answered pretty much all of the question. So I think that is it for now. If the host are present here, Mansoor is here. So I think we can go for concluding the session, or you have the final remarks from the host.
Uh,
Murtaza?
am I, yes, yes, please. Am I audible?
Yeah, you're audible. Yeah, please. I think we are good to go for today. I think so. We have pretty
Yeah,
much answered all the questions. So let's
yeah, sure,
conclude the
sir.
session, please.
Sure, sir.
Okay.
Uh, Thank you so much, sir, for joining us in today's session. And it was really informative and really insightful session. And it's a opportunity for all academia, student, and researcher to connect to each other for a meaningful session and also a meaningful discussion in today's session. And I hope in future we will arrange more sessions about relevant topics, and we will enhance collaborations among the student and researchers and academia. Hope so.
Yes.
And thank you so much
Yeah.
again and again, Dr. Saad, for your time and for joining us. And thank you so much, everyone. And I hope we will to in the field of life science by filling the gap as the professor was telling that there is a gap, a researcher gap in bioinformatics domain, and in future together you students and you researchers will fill up that gap and will make a positive change. Yeah. So at then please give some review. uh, Go ahead, please.
Murza, right now Azan is going on, so you can conclude it for now from my side. Okay, please. Yes. Sorry for the pause. As Azan was going on, so I thought it's better to stop. Yeah. Ultimately, I think Murtaza, I want to say one thing that it's difficult because we have different cohorts of people that are joining us together, and ultimately, what we can do, we can select some cohorts for future studies. But of course, it is very interesting to get on board with everyone. So we are basically a lot of different kinds of people that were joining us today. We have questions from microbes. We have questions from repetitive sequences. Or so. It was a very interesting session. So I think a lot of people will get benefit from this thing, and I would suggest that we should keep this kind of event going on to keep the current alive in the blood of our researchers, our students, so that they get motivated more and more, so that they can learn more and more and they can develop their human resource up to the maximum level. So that's all from my side, guys. Thank you so much once again for the organizers for arranging this event and doing the hard job of basically publicizing it and connecting with you, all of you guys. So keep this up. Big thumbs from my side, and inshallah, in future we will be working together more and more on different events.
How we should
Yes,
like
Mirza.
we should move forward. And
Yes.
thank you so much, everyone. Thank you so much again and again, Dr. Saad, for joining us here.
Okay, Mirza, I will sign out. It's a pleasure to talk to you. Inshallah,
Yeah,
see you
sure,
again soon.
sir.
Okay.
Sure, sir. Thank you so much, sir. Thank you so much, everyone. Have a good night. Allah Hafiz. And must fill up the form, Google form. It's mandatory for attendance, and as a it will be as a certification form. Okay, must fill it.